Good morning. My name is Dan Egel. I am at the Southwest Purdue Ag Center. I am a member of the Botany and Plant Pathology Department here at Purdue, but I am located in Vincennes. I'm located in Vincennes, where one of the things I do is research with high tunnels and greenhouses. I also have extension responsibilities across the state where I'm able to visit a lot of high tunnels. So what I want to talk about today is some of the what I've learned from doing experiments here in Vincennes with high tunnels, and some of the research experience I have with high tunnels. So the title of my talk today then is The Influence of Cultural Practices on Tomato Diseases in High Tunnels. Okay, so first I'd like to go over some definitions. What is a greenhouse versus a high tunnel? Well, technically a high tunnel is a greenhouse without heat. So a high tunnel uses passive heat. A high tunnel is where we plant usually something early in the spring, usually tomatoes, and we count on the sun during the day to warm up the high tunnel, and that heat will last through the night. So technically, if you have a greenhouse, if you have heat, it's a greenhouse. All right. Tomato plants may be grown in the ground or in pots. Both structures protect plants from rain. Both structures, as we'll talk about, increase humidity. Tomatoes are by far, at least in Indiana, are by far the most common crop grown in either structure. If one were to ask me then, what are the most common tomato diseases in Indiana, the slide here shows my answers. Bacterial spot, bacterial speck, bacterial canker. Fungal diseases would be early blight, septoria leaf blight, leaf mold, gray mold, and white mold. So this would be my answer if you ask me just in general, field or greenhouse, what are the most common diseases that you see in Indiana? Now, if you were to ask me specifically, what are the most common tomato diseases in high tunnels, that would leave these diseases: leaf mold, gray mold, and white mold. Well, now I want to talk about why the difference, why some of the I don't see some of those diseases usually in the high tunnels type situation. Most diseases, most foliar diseases of tomato then, or in other plants, most diseases require leaf wetness for infection to start and rain for spread of those diseases. And you see here is a leaf with simulated leaf wetness, and you see the raindrops ahead. And this is what allows diseases to start, and the rain spreads those diseases between plants. Well, when we put plants on a high tunnel, then we've protected them from the rain, and we don't get as much leaf wetness, and that changes the types of diseases that we see on tomato plants. I have a colleague in North Carolina, where they have plenty of heat, who says that a lot of growers down there grow tomatoes in high tunnels for the specific reason that they don't get rain, and it changes the disease situation. So that's great. It helps us that high tunnels protect us from the rain and reduces the number of diseases that we see. However, high tunnels also increase the relative humidity inside that structure. You think of that structure, it's enclosed, it gets humid in there to an extent that it doesn't get humid on the outside, and so that changes what diseases we'll see inside the high tunnel. So, for example, then, this is a graph of relative humidity from midnight to midnight. The blue line you see here is relative humidity inside a high tunnel. This is one of the high tunnels we have here at the farm, and this just happens to be on the 15th of this past August. And then the red line then is the relative humidity for Knox County. So the upper graph, the blue graph then, it shows you that the relative humidity is higher during most of the day, particularly nighttime hours, than the humidity outside in the air. So humidity is higher inside a high tunnel, and that's going to influence the diseases that we see inside the high tunnel. So leaf mold then, leaf mold, gray mold, and white mold, and I'm sorry that plant pathologists weren't able to come up with more creative names for those diseases, but leaf mold, gray mold, and white mold require high relative humidity and are therefore more common in high tunnels than in the field. Usually when I see these diseases, it's in a high tunnel and usually not in a field. Another factor which tends to increase the amount of disease that I see in high tunnels then is tomatoes are often planted on high tunnels, as seen here, without crop rotation. So for example, if one were to have a field of tomatoes, an acre of tomatoes, 1 year, the next year most people wouldn't dream of planting tomatoes in that same spot the next year. However, in high tunnels, because of the value of that real estate then, because it's enclosed, people tend to plant tomatoes after tomatoes every year because tomatoes are a valuable cash crop, and because they need to put them in that high tunnel to increase the season. I should mention that this particular greenhouse shown here, these individuals actually move their greenhouse from place to place every year. Most people don't do that, but that is another option. However, what I'd like to talk about today is ways to get around this crop rotation problem so you can grow tomatoes year after year and minimize the disease situation. So let me just introduce the diseases then. Leaf mold, gray mold, and white mold. Leaf mold, then, if you see a leaf mold, the first thing you'll often see is these kind of yellow or what I call chlorotic lesions on the upper surface of the leaf. You see these kind of yellow, and to me the margins seem kind of diffuse. They're not angular, they don't stop with the vein, it's just kind of diffuse yellow lesions on the top of the leaf. Now if you're to flip that leaf over, then you'll see this kind of gray-green mold visible on the underside of the leaf, and this is really diagnostic for this disease. And if you see the yellow lesions at the top and flip it over and you see this gray-green mold underneath, you can be pretty certain that leaf mold is what you're seeing. Now this photograph here I took this past year in one of our high tunnels, so you can see this disease can get pretty severe if left unchecked. And that's what we did this year. We didn't do anything to stop it. In fact, I wanted to monitor the progress of the disease. So you can imagine the dead leaves seen here will affect the fruit quality and will affect the amount of blossoms that'll occur. Now, this disease does not affect stems, does not affect fruit, but it will affect the quality of fruit and the amount of blooms over time. I want to talk later about some specifics or some general disease control options, but specifically then for leaf mold, this disease is caused by a fungus that survives in crop residue, so you want to reduce crop residue. In the field, crop rotation and fall tillage would be an option. Resistant varieties are an option. I'll talk about that a little bit. And we'll talk also about ways to reduce relative humidity in the greenhouse, because this is a disease that loves high relative humidity. The second disease I want to talk about is botrytis gray mold, or sometimes just called gray mold. And if you look at this photograph, let's see if I can use the arrow here. If you look right along this surface here, and you can probably see that there's actually the fungus is growing on the leaf. So on this, I'll talk about a 10 times hand lens, but I recommend a 10 times hand lens that helps one to identify structures such as these, which helps one to identify diseases. So this is gray mold. This is a more serious lesion. Again, you can see here the actual fungus growing in the leaf, and you see in the background that this is a high tunnel type situation, and again that it's common to see this disease in high tunnels. This disease can also occur on stems in serious situations, and obviously this is a more serious situation than on the leaves. If it's killing the stem, it's killing the plant. This disease will also occur on the fruit, but I don't usually see that. But it can be a serious disease. So gray mold of tomato then affects many different types of plants. So, for example, a lot of ornamental plants are affected as well. So your geraniums and whatnot, if you're growing those in with your tomatoes, that may cause some problems. Keep temperatures up and humidity down. There's been some success liming soils for raising the pH. Keep fertility levels up. And there's been some research that shows that calcium levels should be twice what phosphorus levels are in order to manage grain mold. And again, I'll talk about some other cultural methods towards the end of my talk. All right, so when you see white mold, or sometimes called timber rot, you'll see oftentimes the first thing what you'll see is a plant wilting like this. And you look at that plant and you say, well, what's the problem there? Well, if you look at the base of the plant, what you'll notice is the stem, instead of being green, normal green, it's a brown color. And I think that's why sometimes it's called timber rot, because it looks like a piece of timber. So you see here the brown, kind of light brown stem. The stem has died, so that's why the plant is dying. But we also see in this photograph then there's some dark black irregular shaped fungal structures, as well as some white mold associated with these structures. When these structures, should these fall to the ground, what will happen next year is a very small mushroom will grow, seen here. Very small mushroom. And the spores to this mushroom will land on the plant, and what happens is, especially, they're likely to land on old tomato blossoms and get into the tomato in that way, and grow within the plant and often end up killing the plant. This can be very serious disease. So white mold then has a very large host range. Unfortunately, it affects pretty much any broadleaf plant. Certainly, any kind of ornamental plant can be affected. I've seen it on cucurbits. It can occur on soybeans even. So, and it lasts a long time in the soil. So crop rotation is unfortunately not even a real good option for this disease. You want to keep humidity levels down, because when those spores in the spring land on those tomato blossoms, if it's humid, then that makes it more likely that the infection can occur. I don't want to spend a lot of time talking about it today, but there is a biocontrol option known as contans. And if you know you have a problem in your high tunnel, and if you rototill that in the fall, there has been some successes with that biocontrol option. And in fact, that's organically certified as well. All right, now I'd like to go ahead and I've talked about specific diseases. Now I'd like to talk in general terms about what we can do to minimize diseases in high tunnels. All right, so I've talked about reducing crop residue, and of course, if you can move your fields, your high tunnel crop rotation is certainly an option. But if you can't rotate your crop, what I'll talk about is removing the crop entirely from the high tunnel, removing the entire plant as much as possible from the high tunnel, so that residue is removed from the greenhouse at the end of the season. In fact, that's what we're doing this late summer period. And then I'll show some photos, but I recommend a floor covering to also minimize the amount of crop residue getting in the soil. We'll also talk about increasing aeration amongst the plant. We'll talk about correct spacing, and I'll show a little bit of data that we've gathered. Prune lower branches. This is especially true with indeterminate plants, but in the most cases, I think what growers are growing, tomato plants are growing staked determinate varieties. And then we'll talk a little bit about ventilation. Okay, so first, greenhouse floor covering. So without crop rotation then, crop residue tends to accumulate in the soil. So if you're growing tomato after tomato, that's what happens. That's the crop residue that accumulates in the soil. And a lot of plant pathogens like the fungus that causes leaf mold and gray mold, may survive in crop residue. So if you have a floor covering, then that tends to reduce the amount of crop residue that gets in the soil. So here's a photograph of our high tunnel this year. This is tomatoes in high tunnel with our floor covering. You see there's staked tomatoes, determinate varieties, and you see the black plastic mulch. In between the rows, you'll see a covering. So if leaves fall on this covering, they can be swept up. If fruit fall, they can be swept up. If you need to remove a plant either during the season or after the season, that residue ends up on the floor covering and doesn't make it into the soil, and so that means less disease next year. This is important for us because along with growers, other growers, I plan to plant tomatoes in these structures next year, and I want a minimum of disease. Okay, so your the greenhouse floor covering that you just saw in the last photograph then is a white woven ground cover. We clean and sanitize it at the end of each year. This is the third year we've used it, and we put it away already. So we can use this year after year. I don't know how many years we've used it, but 3 years seems to work out fine, and I plan to use it again next year. The 800-foot strips that we use, 3 feet wide, cost about $330. But again, I can reuse that. Now underneath this, now I'll show a photograph in a moment here, but underneath that we have a black landscape fabric. We only used that 1 year. That's under the ground cover, and that cost us about $90 per structure. And again, we'll throw that away at the end of the year. So here's a picture of the black landscape fabric. This rests on the ground and just under the white woven ground cover that we saw. You can see my hand through it. It's a rather thin fabric. If you were to walk on it, you without the covering over top of it, you would probably break it and tear it. So that's not enough to put down by itself. So we put this white woven ground cover over top of it, and then it's plenty sturdy enough and protects the ground and the landscape, that cloth underneath it. So then what I show here is a photograph on the left of a high tunnel without any floor covering, and on the right a tunnel with floor covering. And let's imagine that you have in the middle, then this is a tomato with white mold, and you want to remove that plant from the greenhouse. Now imagine doing that in the left-hand house without a floor covering. Those little fungal structures are going to fall on the ground. Tomatoes are going to, or mushrooms are going to grow from those and infect your tomatoes next year. Whereas if you do it on a greenhouse with floor covering, I think what you'll see is those, if they do fall from the tomato plant, they can be swept up easily, and you can sanitize and keep some of that crop residue from hitting the soil and causing a problem next year. This is a photograph I just took the other day. We're cleaning out our greenhouse. We've removed all the plants from the greenhouse. You see the stakes are still there. We haven't removed them, and what you're seeing there is the black landscape fabric. We've removed and sanitized the white covering, but you can see we haven't removed the black landscape cloth yet. But just the point I'm trying to make here is that we're removing the plants entirely from the greenhouse. Another cultural control that you can use then is keeping the area around the high tunnel clear. This will reduce insect pressure. For example, if there's plants growing around the high tunnel, weeds and even ornamental plants, they can harbor insects which can then move to the high tunnel. There's also fungal diseases that can be harbored by plants near the greenhouse as well. And of course, if that area is clear, that means you're going to get better ventilation, because you'll get breezes blowing through the high tunnel when the sides are up. So this is a picture of our high tunnel, and you see there's an area around that we put a ground cover and then gravel over it. So this is free of plants, free of insect pests. You imagine those tiny mushrooms I showed you for white mold. those can't grow too close to the greenhouse in this type of situation. So this is, I think, probably an optimum situation for reducing diseases that might otherwise get into the high tunnel or greenhouse. Okay, we've talked about ventilation. In a greenhouse type situation, growing things in the spring, the early spring, what you would do is open those vents, let some in the evening, let some of that drier, cooler air into the greenhouse, replace the humid air, close the greenhouse up, turn the heaters up to heat that air. Okay, so that's what you do in a greenhouse type situation. In a high tunnel where that's where the heat is passive, sometimes you can't do that. Obviously, sometimes you need to maintain that heat and not ventilate to keep those houses warm over the night. But when temperatures warm up and you can ventilate, we can't open the sides, maybe you can open the top or whatever you can do. You want to ventilate those at night to replace the humid air and allow as much ventilation as you can. Obviously, when temperatures are very cool, then you need to use common sense and not open it up when you're going to harm the plants, but ventilate and open up the house when you can. Plants may be pruned in some cases to increase ventilation. This is especially true, as we mentioned, with indeterminate tomatoes, which can be pruned at the bottom. And avoid crowding plants. It's been my experience when I go in a house, I think sometimes people are putting too many plants too close together, both within the row and especially between rows. This is a picture of a greenhouse, not a high tunnel, but you see the fan there. In some cases, it's possible to have fans that will move the air around and in general help to keep the air less humid. I'd like to change gears here. I've talked about some diseases and some cultural practices. Now I'd like to talk about some an experiment that we did this year, and we're just kind of starting to go through the data. As I mentioned, we're still cleaning out the plants themselves, but I want to talk about an experiment I did to try to look at plant spacing and what the optimum is in a high tunnel situation for tomatoes. Okay, so the plant population study we did then, we had 5 spacing treatments that we did, and as you see, the left-hand corner here is, for example, this is 16 inches. Tomatoes were in 16 inches within the row with what we call a Florida weave. I'll show a photograph of this. 20 inches within the row, Florida weave. 20 inches Spanish trellis, and I'll show the difference between a Spanish trellis and a Florida weave in a moment. And then 24 and 28 inches within the row, Florida weave. Those were the plant population, the spacing treatments. Then on this side we have 2 different tomato hybrids. Mountain Spring, you can see here, is a variety I chose because I know it's very popular among growers, lots of tomatoes in that kind of mountain series. And then Red Deuce is the other tomato I'll choose. And I did this because Shuben Saha, who I'll talk about in a moment, who did experiments before I was doing them down here. I had excellent success with Red Deuce in a high tunnel type situation. All right, so our study then, our tomato population study then, we moved the tomato transplants into the high tunnel March 14th, and our last harvest was September 11th. So that's a nice long season, and we'll see some data for that. We fertigated 4 times a day in the main season. We started off 3 times a day, but moved to 4 times a day in the main season. So day in and day out, they were fertigated every day. So that means water and fertilizer included with it. That's 120 gallons per day per high tunnel, and each high tunnel has about 420 linear feet of tomatoes. The primary fertilizer we use, this is varied a lot, but the primary fertilizer we use, potassium nitrate, shown there, 13.5-0-46.2. We collected fruit weight and fruit number. The major insect damage that we saw was stink bug damage, and the major horticultural problem we saw was zipper scars. Now then, this is a photograph of the greenhouse, and what I want to show here, first, let's mention that I had this on 5-foot center. From center to center is 5 foot here. And I wanted to do an experiment where I experimented with closer spacing between the rows, but that didn't prove practical. So for that reason, I adjusted spacing within the row instead. This first system is called the Spanish trellis, and you see here there's 2 posts on either side, and the string goes like this, kind of forms a cage, and then the plants are brought up through this, and then this is tied periodically as the plants get taller. This is the Spanish trellis system. Then over here we have the more typical Florida weave, where you have the single posts, and the twine is woven among these plants, and this is fairly typical. All right. So one of the things that I thought would happen then is that we would have, the closer we space the plants, the more disease you would have, and that's not really what happened. The disease that happened to show up is leaf mold, and that's so you see on this axis here disease severity. The higher the bars, the more disease, and then shown along this axis are the different treatments we talked about, where the numbers are spacing within row spacing in inches, and then weave is a Florida weave, and trellis is a Spanish trellis. There's looks like some differences here. As you can see, it looks like the 20-inch weave has more disease than the 20-inch trellis, Spanish trellis. But in fact, there's no statistical difference here. That's why there's no letters shown here. So I did not find any differences within the mountain spring. Now this is the blue is the mountain spring. No differences between these different treatments. However, it is significant then that the red deuce showed no disease, absolutely no disease on the red deuce throughout the entire season. So then, leaf mold severity was not affected by in-row spacing. I was not, again, able to measure this is between rows. I wasn't able to tell the difference between, say, 4 and 5 foot rows, but leaf mold severity was not affected by in-row spacing. There may be other diseases that are affected by spacing, but at least this year I was not able to show differences in leaf mold severity by in-row spacing. Red deuce was resistant to leaf mold in this experiment. I did not see any leaf mold on any of the red deuce variety. That's interesting because red deuce is not listed as resistant, so that surprised me. I've read that people don't a lot of people don't put much stock in the resistance rating for leaf mold because the fungus can be so variable. So you could plant red deuce next year and have different results. But this tells me that there are differences out there, and if one should notice the catalog, whether they're listed as resistant or not, and should also notice one's old results with diseases in high tunnel situation. Now, my second data slide here, what we're looking at is we're looking at yield. So the higher the bar, the more the yield. And again, red is red deuce, and the blue is Mountain Springs. And again, we have over here the spacing in inches, and trellis is Spanish trellis, and then the weave is the Florida weave. Well, there's no numbers here, no letters listed here. So basically, what I'm showing here, you can see slight differences in yield. There were no differences in average yield of tomatoes based on in-row spacing. No differences in yield. So that kind of surprised me. But the weight of tomatoes per plot was not affected by in-row spacing. So that's I'll continue to analyze data, but at least at this point, that seems to be one of my conclusions. Now, Let's look at number. The number of tomatoes produced. We looked at weight, but let's look at number of tomatoes produced. So the higher the bar, the more tomatoes produced. Again, the blue is Mountain Spring and the red is Red Deuce. Here's our treatments here. There were significant differences here. So you see if 2 bars share a common letter, in this case A, between the 16-inch Florida weave and the 20-inch Florida weave, in this case there's no significant difference between these. But you can see that there are some differences. So here the 24-inch Florida weave Mountain Spring has significantly less tomatoes than the 16-inch Florida weave. What you'll notice is that in general these treatments with wide spacings, 28-inch weave, 24-inch weave, 28-inch weave, these spacings tend to have fewer tomatoes, whereas the closer spacings like these 16-inch weave tended to have a higher number of tomatoes. In conclusion, then we might say that the closer the tomato are spaced, the more tomatoes we had. And then we looked at average fruit weight. This is the size of individual fruit now, and we see that again this 28-inch weave where we had the plants further apart tended to be, the fruit tended to be larger than when the plants were put closer together. So this kind of makes sense to me. Also, it's interesting, the Red Deuce fruit tended to be larger than the Mountain Spring fruit. So then the conclusions here then are rows with more plants, that is closer spacings, had more fruit. The closer spaced fruit had smaller fruit. So we can get more fruit off of those plants that are close spacer, but those fruit tend to be smaller. And also another conclusion is that the Red Deuce had larger fruit than the Mountain Spring. Alright, so now what I'm looking at is just overall what happened during the season, tomato yield in pounds, marketable tomato yields, and you see again the Mountain Spring and the Red Deuce, and we planted in March, and you see the yield steadily going up till in July 9th here we're getting a large amount of fruit, and then this tends to go down until the end of July, beginning of August. We have this kind of period here where we didn't get many fruit, and it might have been tempting to tear those plants up, but we had a second, what I'll call a flush, and the end of August being in September before the plants started to go down again. So I don't know what your markets might be for this second flush, but it's interesting to know that for both the Mountain Spring and the Red Deuce, we did get a second flush of tomatoes later in the summer. That's marketable yield. The same is true for number of fruit shown here. Again, the higher the bar, the more fruit, but we tended to get this early flush, our main season, and then get a second season of tomatoes later in the summer. So that's something to think about. It might be tempting in late July to tear those things up, but you need to kind of look at your market and see if that's going to be worthwhile. Alright, I want to kind of dispense with the data at this point and encourage growers to scout for problems in high tunnels, like this young lady is doing. She's got a hand lens looking at the leaf. I encourage you to, whether it's photographs or looking through the hand lens, and you may decide to contact somebody like me. I send samples up to our excellent diagnostic clinic on campus. But let's find out what those problems are now during the season, so that you can think about this and talk to somebody like myself about what to do about those problems next year. So scout for problems in the high tunnel while those problems are there, and let's find out what those problems are. I also wanted to point out that now this year Purdue has its own Tomato Doctor application. Works well, I know, for Apple products, for the iPad and the iPhone. I know it works real well. So this has nice photographs of the problems, and it also has descriptions of the disease. So I encourage you to take a look at that. Now, before I go any further, this has been a tremendous amount of work this year for this experience, so I want to just mention some acknowledgments. So shown on the left here is Shaben Saha. He was at Purdue and really started the high tunnel program there, and so I'm just learning kind of from him. He's at the University of Kentucky now, but we're in close contact, and so I want to give him credit for starting that program and for teaching me so much about it. Dennis Nowaski's in the middle there. He's a superintendent. He built and maintains a high tunnel, and I certainly couldn't do these experiments without Dennis. And then, of course, there's me on the right. Sarah Hoke is my technician and assists with harvest and data. She wasn't involved to a large extent with high tunnels, but she certainly helped do a lot of things during the year. But especially, I want to thank on the left Larry Sutter. Started off, learned a lot of tools of the trade from Shaben Saha when he was here, and he was in charge of the high tunnels in 2014 and did everything I asked him to do, and then some. And then Michael Russell on the right just finished school at Ball State, has a degree, and probably going to look for another job, and I'll be sorry to lose him, but he's been a tremendous help. So I couldn't do these experiments without these individuals. So with that, I'll conclude here. If there's any questions now, or if somebody wants to contact me in the future, I think my email was at the beginning of the presentation, I'll be glad to answer any questions.